So hello everyone, welcome to my channel Simply Vinay. So today we are going to talk about the new biotechnology book that has been recently launched from the NCERT, and UPSC and other exams is constantly asking from this syllabus. Okay, so this is a different book from our class 11 normal textbook, because in this so what happened is that in these the chapters like this chapter and this chapter and the ninth and 10th chapter were now advanced and were asked and were given in that book. So if you want to do well in the biology, then you can watch this lecture. This lecture is useful for the CAPF assistant commandant, CDS, NDA and BPSC exams, where you have a good proportion of the biology questions, because if you see the biology trend, it is going from the 9th and 10th NCERTs to the 11th and 12th NCERTs, and especially on the 11th NCERT. So let's get started. So this is our syllabus. So we have to read this chapter 2. Each and every topic, each and every topic you will find in the 9th and 10th NCERT, but here it is given in a more comprehensive and a more advanced manner, from which the recent questions have been asked. Okay, then after we will proceed for the biomolecules and enzymes and bioenergetics. So these 4 chapters are very important. You can read this chapter also, cell cycle, but I think the first chapter 2, chapter 3 and chapter 4 are the most important. So let's get started. So first we will I have already read it, so I will only tell you the facts and figures so that you can either mark or make a short notes over it. So let's get started. So this is the introduction chapter. So here I am telling you what things you have to make notes or make facts. Okay, so if the questions comes that who was the scientist who discovered that thread-like structure bodies during the cell division, that is what we see like a gene or a fiber, then his name is Walter Fleming. This question can come. So the next, who clicked that we all know this question, that who proposed a double helical structure of the DNA, which we see of the double helical structure, that is James Watson and Francis. So this is a very important topic. From previously the UPSC have asked around 4 to 5 questions from these 4 lines only. So what are these lines? These are the colors of biotechnology. So here it is divided into 4 colors. Okay, that is the blue, green, red and white. So what do you mean by blue biotechnology? This is the application for the application of biotechnology. All these are the applications of biotechnology. Okay, so blue you can relate to the marine life. So this is the application of biotechnology for the marine and the freshwater organism, and also in developing new drugs. See, if it comes that which biotechnology is useful for developing new drugs, we will always mark the white, since drugs are white and white technology sounds comparable. But no, we have to see in the depth that blue biotechnology, it's for marine and freshwater organism, that's okay. Seafood, okay. Dangerous waterborne disease, okay. But developing new drugs, this is also comes in the blue biotechnology. In the green, environmental friendly solutions and plants with improved traits, like we have like the gene therapy or the BT cotton or the genetically edited rice and other crops. So now for the red biotechnology, it is, see, this is the most important. The rate we cannot relate that it is related to the pharmaceuticals product, like production of insulin, enzymes, antibiotics and vaccines. This is a very important thing. Now for the white biotechnology, it's for the industrial purpose, that the use of enzymes as industrial catalyst in eco-friendly manner to production of valuable chemicals. This is the main line, I think so. Okay, the production of valuable chemicals using some enzymes and other things. Now we move forward. So this is the first successful report of the gene therapy was achieved when by D. Silvia. Okay, so now next, this is the biobutanol. Thanks to our road ministry, who is always talking about the petroleum, that is the biobutanol, who in the diesel they want to put bioethanol in the diesel. So bioethanol is a fuel that is often replacement for the gasoline, and gasoline is a, like, see, it is already very common in the as a biofuel in Europe and others. Okay, so these are two facts, that the Serum Institute come first, then the National Biotechnology Board was established. The Serum Institute was established in 1960s and Biocon in 1978. These are the topmost institutes in India which make the vaccines for India and all over the world in a very less price. Okay, so if there comes in a chronology question, always remember the Serum Institute come first, then the Biocon, and then the National Biotechnology Board. So this is a simple fact. Let's move forward. So this is the end of the chapter. Now we are starting with our cell organelles, and this is the most important, very important. Okay, so let's get started. We are starting with the overview. Let we all know our first topic, the plasma membrane. If we have a cell, the outermost membrane is the plasma membrane, or also we known as the cell membrane. So first, it is semi-permeable in nature. The first fact, it is we all know that it is semi-permeable in nature. If it will be fully permeable, the cell will not exist, and if it's fully rigid, then the exchange of the gases and the liquids will not happen. Okay, so our next fact is that who get the fluid mosaic model. So I have not read too much about this model, that because it's very depth model who is coming in the NEET. So I just that who have given this. The fluid mosaic model is given by Singer and Nicholson. Remember these two names, like Singer and Nicholson give the fluid mosaic model. This is the main thing. Okay, now these things, that the lipid bilayer makes the cell boundary is in a quasi-fluid state, and this is dynamic in nature. So who is responsible for making the cell outer membrane as a dynamic and semi-fluid? That is the lipid bilayer membrane. Also, they are made up of the hydrophilic head and hydrophobic tail. So remember, just like a sperm, this here the head is hydrophilic and the tail is hydrophobic. The meaning of these things, that the cell membrane is also made of like fibers, small fibers, where which have a head and a tail. So the head is attracted to water and the tail is hydrophobic to water. That's the main thing only. Just remember this, like a sperm. Okay, now see, these are the main things. So what do you know mesosomes? So mesosomes are like extensions. Mesosomes are like the extensions of a plasma membrane. There is a special membrane structure is formed by the extension of plasma membrane, also known as mesosomes. So this is a simple fact. For our competitive exams, we have to just read the facts and figures and know. The main thing about this book is that whenever a new term comes, they give it in a bold sentence. And on analyzing the last 5 years of the biology questions, I have seen that if something is marked in a bold manner, UPSC asks, or any competitive exams in which biology comes, they ask that what is so mesosomes is simple extension of plasma membrane. Okay, so what is the use of mesosomes? Increase the surface area of the plasma membrane for the gaseous diffusion and other things that you can tell. So now see, we all know that it is semi-permeable in nature. That means it is used for the transportation. So there are two types of transportation here, that is the active transportation and the passive transportation. So the passive transportation occurs by two things, that is the diffusion and the osmosis. Okay, so we all know that the diffusion and what is the osmosis? Osmosis is with respect to the concentration, and diffusion is with relation to the same with the permeable membrane. Okay, now see, this is a PYQ. What do you mean by aquaporins? So simple thing is that aquaporins are one of the critical channel protein for transportation of water in the plants and animal cell across the plasma membrane. So what is this? Aquaporins are like a small channel in the cell membrane. So if we have a cell wall, then there are small channels. This is the aquaporins. This is a site for the exchange of water. Remember this, aqua means water. Okay, so in both plants and animals, remember this. And with the help of ion channels. The mean of ion channels is that the canal is not always open. Whenever a specific type of ion comes, this channel get open, and if not, it get blocked. So this is also a main point which I am marked very important, like this. So here it is told that it is worked on the ion channel. So how it works? So there are two type of, we have here learned there are passive type of two types, diffusion and osmosis, and here we are learning about the active transport. Active transport where means where the ions are involved. There are two types of ion that are mainly responsible for this, that is sodium ion and the potassium ion. Okay, now see, in if the transportation, these are mainly of the transportation. So there's things written here that in a coupled transport, if the molecules are transported in the same direction, that is uptake of glucose and sodium. That means if the suppose the glucose is coming into the cell and the sodium ion is also coming from the same direction, that is inside only the cell, then it will call as symport. Okay, and if the transportation of the two molecules involve in opposite direction, that transportation of Ca and calcium and sodium and calcium by the Na or Ca plus 2 are antipoters. That means if the nucleus is coming in but the sodium is coming out, then it is known as antipode. So this is a main thing. And when, this is also a main point, that is what is unipode. While facilitated diffusion transport only single molecules, for example glucose, it is known as unipode. So if there is a transportation of only glucose, no ions is involved, then it is known as unipode. Okay, now we are moving toward the cell wall. So what is the difference between plasma membrane or cell membrane or the cell wall? Both first two things are same, and the cell wall is only present in the plant cell. It is not present in the animal cell. So it is written here, the cell wall is cell of the bacteria. It is present in the bacteria, algae, fungi and higher plants. So remember that the bacteria also possess cell wall, fungi always possess cell wall, algae always possess cell wall. So remember, except the animal cell, all the organism, whether it is a bacteria, algae, fungi, anything else, because there are a lot of bacteria, there are a lot of algae, so they possess the cell wall. So in bacteria, this is a very important thing which has been recently asked in one of the PYQ in UPSC, that in a bacteria it is composed of the polysaccharide cross-linked by small peptides. That it is written that the cell wall of bacteria is made of what? It is composed of polysaccharides of cross-linked by small peptides. Okay, and in the eukaryotic it is primarily made of the polysaccharides. So what is the difference? So the difference is here. Both are made of polysaccharides, that's true, but what is the difference? The difference is that it is cross-linked by small peptide bonds, and there it is just normal with the normal size bonds. Now there are two types of the bacteria. They are gram positive and gram negative bacteria. So it's a very simple difference. I am telling you that positive means thick. So there are two types of bacteria, that is the gram positive and gram negative. Positive means thick, negative means thin. And there are only 2 points to remember. There is single membrane, one membrane. It's written that it is a single plasma membrane. So remember, there is one plasma membrane, and here point number 2, they have two plasma membranes, that is one cell wall, one plasma membrane, then another plasma membrane in the gram negative bacteria. Okay, also one more very important points, that when see bacteria, when we get ill, doctors give it antibiotics. What these things do, they go inside our body and kill the bacteria. So the gram positive bacteria are killed by the antibiotics, but in the gram negative they are not, they are no effect. Antibiotic work here and antibiotic don't work here. This is a mainly simple difference. And if you ask that why the antibiotic work here, see the antibiotic goes to the cross linked small, it is written here that they are cross linked by small peptides. So what the antibiotic go and they stick to the cross link and destroy it. When the link get destroyed, they don't get food and ultimately get killed. So this is the main thing. So the antibiotic attack on the cross linked. So it is a fact that cell wall is made up of cellulose, which is a linear polymer of glucose residue. That is, cellulose is a simply a glucose. Okay, whereas in the fungi it is made up of chitin. Now another things, what do you mean by lignin? So a secondary cell wall is rigid and thick compared to the primary cell wall due to the deposition of lignin. So lignin is a type of layer which is on the secondary layer, which makes it what? Rigid and thick. So this is the main thing about the lignin. Also read one more this point. What is plasmodesmata? So it is a layer of calcium pectate holds together neighboring cells and their cytoplasm together, a structure called plasmodesmata. Now this is a very important point. What is glycocalyx? So in prokaryotic, especially in the bacteria, the cell is further covered in a heavily glycosylated protein known as glycocalyx. So what happened, the bacteria is a prokaryotic organism, that is they have a non-defined nucleus. Okay, so what happened in these the cell wall, the outer covering has a special liquid known as glycocalyx, which give its further strength. This is the main of the glycocalyx. So glycocalyx is simple liquid in the prokaryotic bacteria. Remember these things. It is in the bacteria, and which type of bacteria? That is prokaryotic bacteria. Now we are moving to our next topic, that endomembrane system. So what is the endomembrane system? Endomembrane system are simply the organelles which have a membrane bound structure. So like the prokaryotics, who do not have any well membrane, like see, this is a cell, we have different organisms, or we can say organelles. The organelles is in a fixed membrane, they will have a fixed size, they will have a fixed position, then these are known as endomembrane system. So moving forward, these are the examples of the endomembrane system, that include endoplasmic reticulum, Golgi complex, lysosomes plus vacuoles. These are the endomembrane system, these four. So we will read all these in a depth manner. Now our first topic, the endoplasmic reticulum. So it is located near the nucleus and the Golgi apparatus. That sound fair. The main thing is that whenever we are talking about any of the cell organelle, first remember its function, second its location, and third its use in cell. What was its use, and fourth if any special type of quality, something special, what is something special about it. So these four things are for the every organelles. So first thing, endoplasmic reticulum is exclusively present in the in the eukaryotic cell. So this type of things, that is accept, exclusive, these are the UPSC favorite questions. So it will tell a endoplasmic reticulum is both in the prokaryotic and the eukaryotic. No, it is only in the eukaryotic, the first point. Also it is a it's involved, we all know that it poses the rhizobium, which is for the protein synthesis and the storage and the lipid. So there are two type of endoplasmic reticulum, rough and smooth. See, this is a type of structure you can imagine. There is a nucleus, there is a rough, then beside it is a rough endoplasmic reticulum, and same here there is a smooth also. Then a little far there is a Golgi apparatus, then the vesicles, and there is a cell exterior. Imagine like this: nucleus, endoplasmic reticulum, Golgi apparatus, vacuoles or vesicles, and then the cell exterior. So what are the rough endoplasmic reticulum? They are the protein synthesis. Presence of the rhizobium on its cytosolic surface. They are for the protein synthesis. The first point. There is only one thing, that it is on, that is for the protein synthesis. The second thing, they are it's a smooth endoplasmic reticulum. So all lipids are hydrophobic. Remember this. They are hydrophobic, that means they are insoluble in water. This is a previous year PYQ. Okay, so remember this. And they cannot be used in the synthesis of cytosol. Okay, so the synthesis of so what are the smooth endoplasmic reticulum produce? The rough produce the protein. So what is the smooth produce? Smooth produce the lipid plus fats used for the cell. So it is made up of the phospholipids. It is also written that a phospholipid is one of the crucial component of a membrane. Okay, this is a very important point, that is smooth endoplasmic reticulum is an essential fight for cholesterol synthesis. So if the UPSC comes that what is cholesterol, there is a previous PYQ of the CAPF. What is cholesterol? Cholesterol is a lipid. Who produce the lipid in a cell? Smooth endoplasmic reticulum. This is the connection you have to make. Now we are moving towards the Golgi apparatus. So Golgi apparatus is just like a transportation body. It's just like you imagine it is a road network. It's only used for the transportation purpose. There is no use of anything else. Okay, so first remember what I am telling. The Golgi apparatus. You see here, the Golgi apparatus is of two sides. There is a nucleus, there is a cell wall. Okay, now Golgi apparatus is of structure like this. Okay, so what do you mean by this structure? They, first of all their folds are parallel. They are all parallel. Second, their nearest fold to the nucleus is of convex nature, that is like this, moving towards the nucleus, and their end towards the cell wall, it's of concave nature, that is bend inwards. It's bend outward, it is bend inward. This is the main thing. So Golgi apparatus is its structure is very famous. Its structure is very famous. It is in a type of stack which are parallel, and its structure is known as cisterine shape. Cisterine shape is like this. Okay. So the phase which is near nucleus is known as cis. The cis phase nearest to the endoplasmic reticulum, and the trans phase is from the away. So the cis phase is convex and the trans phase is concave. Remember this. See, this is a very important point, that a protein synthesized by the ribosomes by the rough endoplasmic reticulum are modified in the cisterine of the Golgi apparatus before they are released from the trans phase. That means remember these things, that the Golgi apparatus has two phases, cis and trans. Cis for intake and trans is for outside. That whatever the endoplasmic reticulum make, they give it to cis, and cis pass it to trans, trans pass it to other bodies. Now we are reading about lysosomes. So what are lysosomes? Lysosomes are like spherical vesicles, okay, and they contain of hydrolytic enzymes. Hydrolytic enzymes are acidic enzymes which are capable of dissolving the cell inner organelles. Okay, so lysosomes carry out intracellular digestion. The first point we all reading since the 9th-10th chapter. They possess the hydrolytic enzymes which are produced from the smooth endoplasmic reticulum. Remember this. So they recycle the cell own organelle matter through a process called autophagy. So remember this. If the questions come, what is autophagy? Auto means automatic, okay, phagy means recycle. So which cell organelles automatic recycle? That is the lysosomes. Next topic, vacuoles. So vacuoles are covered by a membrane called the tonoplast. Whenever we are reading about the cell membrane, we see a large vacuole. The vacuole outer membrane is of tonoplast. Simple, 1 point over. Now we are reading about mitochondria. Mitochondria are sausage shape, okay. Remember first point, mitochondria are sausage shape. They are rod structures or cylindrical, like this, okay. They are double membrane, okay. They contain the phospholipids bilayer with protein, okay. The outer mitochondria has two layers, inner and outer. The outer is smooth, remember these things, but the inner has folding. This is a previous year PYQ. They have made it opposite, that the outer is rough and the inner is smooth, but here it is opposite. The outer is smooth and the inner is folded, so that's why it looks rough. So the outer membrane is smooth but the inner membrane has folding called cristae, okay, which provide this large surface area. So mitochondria contains the DNA, ribosomes and few RNA molecules. So remember these things. It is contain a liquid known as mitochondria matrix. Where now we are talking about the plastids. So plastids are like a small box which store different types of chemicals which gives the color to the plant. So there are three types of colors which can be given in this box. That is the chloroplast, chromoplast and leucoplast. Okay. So chloro means green, chloroplast gives a green color, the green color liquid in the plastids. Chromoplast is again divided into two types, the carotene and the xanthophiles. If I'm not wrong, the carotene gives us, I think, gives red and orange color, and xanthophiles give the yellow and other colors. Okay, so leucoplast is just like empty box which is used for the storage of various kind of foods, and it stores three kind of foods: the alveolar plast, which is for the storage of proteins; amyloplast, amylo means it stores starch; areoplast means store protein; and elaioplast or lipoplast, which is for oil. So if you see so if there is I in the name, that means it store the oil. Okay, so and if you see there is a U, areo, then it go for the protein, and if it's amylo, then for starch. Okay, now moving to the next topic, that is the chloroplast. So chloroplast first, they are its location where is mesophile cell of the leaves. Okay, so they are in the leaves. They are of oval shape, as we can see here. The largest organelle in the plant cell. Very good topic. Simple fact come in the exam. Which is the largest organelle in the plant cell? We will not remember that it is a simple chloroplast. Also, there in the chloroplast there are, see this, a chloroplast is bounded by double membrane, and third inner membrane, which is known as thylakoids. Okay, so the thing is that this is a chloroplast, and it is, and the membrane is no, see this, these are thylakoids. So thylakoids are like inner stacks like this. Okay, so thylakoids are arranged in a stack honor. So remember this diagram. What is thylakoid? Thylakoid is a organelle inside the chloroplast, and chloroplast itself is the largest cell organelle. Okay, now we are proceeding with our microbodies. So one important point here also, the microbodies are single membrane, and second, they are only present in the eukaryotic. Remember two things, that if you see any of these names, that is the paroxysmos and the glycosomes and peroxomes. So if you see this name, remember they are only single membrane and only present in the eukaryotic cell. So the first point, the peroxomes. So they are site of enzymes where they are involved in a metabolic reaction. Simple, it is a site. It is a small organelle which is involved, which is used as a site for enzymes to involve in the metabolic reaction. Its function: oxidation and lipid biosynthesis. These are the two major functions of this, that is oxidation and lipid biosynthesis. Our next, glycosomes. So glycosomes are specialized peroxomes found in the fungi and other plants. So remember this, glycosomes are none other than specialized peroxomes which are found in the fungi and plants. Remember this. And work same, oxidation and lipid biosynthesis. Okay, so that's all for today. In the next topic we will read about the centromes and centrioles, which are specific to the animal cell. Thank you, have a good day.